Good morning, everyone. Let's get started. Okay, so, oops. Okay, so before the first midterm, we talk about the genes, two genes that are located on different chromosomes. All right, and because of the Mendel's law, so after the meiosis, you're going to see different genotypes that are different from their parental type, right? So the progeny will carry a different combination of the alleles from their parents, and we say that kind of phenomenon is the recombination. So the recombination actually means you see different combination of the alleles from their parents. All right, because they are located on different chromosomes. So as we all know, they are sorted independently. Then from the progeny, so you're going to see 50%, half of the progeny will have the same genotypes as their parents. So those are the parental type, and the other half, 50% of the progeny will have different genotypes. So we say they are recombinants. All right. So before the midterm one, we focus on this. The genes are on different chromosomes. So from now on, we are focusing on genes that are located on the same chromosome, like this, right? Oops, like this. So as an example here, so you can see 3 genes, A, B, and C. They are on the same chromosome. In this case, we say these 3 genes are linked. They are linked because they are on the same chromosome. And so those linked genes can also produce recombinants. So which means like even these 3 genes are linked. So during the meiosis, after the meiosis, you still can see different combinations like this in the progeny. So those are different from their parents, right? Those are the parental type. All right, so what caused this recombinants? So this is our focus now. So from now on, we're gonna focus on the crossing over, because in this case, the linked genes can produce recombinants through crossing over here. So I hope everyone knows this. All right, because I do not have time. We do not have time to go over this in detail. So when you have time, please read textbook to understand, to make sure you understand that the crossing over occurring during the meiosis. This involves the DNA break in the DNA and the rejoin, and then you're going to see the DNA swap between these 2 homologous chromosomes. All right, so this is our focus now. All right, okay, so I hope you understand the difference between these 2 situations. Okay, so this is before the midterm one, and this is now after the midterm one. So we previously focused on the genes that are not linked. They are located on different chromosomes, right? So this one and this one. Okay, and now we move forward. We now study linked genes which are located on the same chromosome. So this is our focus now. All right. Okay, another fact I would like to mention now is, okay, so we're going to talk about this more in a few minutes. So if you compare these 2 situations, as you can see, so when the 2 genes are independently assorted, the recombinants from the progeny will be 50%, exactly 50%. However, if genes are linked, so the recombinant from the test cross will be less than 50, will be significantly less than 50%. So this is the fact. I hope everyone can get this fact. So we're going to talk about this later. All right, now let's learn some notations and the terminologies here. All right, so so there's a term, so there's a concept which is very important now. So I will write it here. So phase. So phase means the arrangement of two alleles, right? So let's look at here. So suppose, so this is a diploid individual. So in each cell, we have two homologous chromosomes, right? So for each gene, we have two copies, right? One allele, one allele, one allele, one allele here. Okay. Now, if we focus on the capital A and the capital B, if the big A and the big B are on the same homologous chromosome like this, all right? So if big A and the big B are on the same homologous chromosome like this, we say the phase of A and B is cis, right? So this is the cis phase, and we can use this notation to represent this, right? We use, so this is a little bit different from before. We use a slash to separate this two homologous chromosomes. And you're going to see the big A and the big B on one side, the small a and the small b on the other side, right? So this is the new notation. If the phase of AB, big A and the big B, little a and the little b is known, so we can use this notation. So on the contrary, if the big A and the big B are on different chromosomes, so in this case, we're going to say the phase, their phase is trans phase, right? And we use very similar notation to represent this. So this is the new notation we're going to use from now on. Okay. Now, when we look back, so in previously, when two genes are independently assorted, which means they are two genes are on different chromosome, then if we know this, if we knew this, so we're going to use the semicolon to separate this. This means they are independent, so they are not linked. All right, but sometimes, yeah, if you do not know whether two genes are linked or not, so if this is the case, you do not know their linkage situation, so you use a dot to separate these two genes. So then we can perform a test cross to figure out whether these two genes are linked or not. So this is what we're going to learn today. All right. Okay. So, all right. So genetic mapping. So what is genetic mapping? Oops. Oh, it's here. So what is genetic mapping? So first we knew. First we know like some genes are located on the same chromosome, right? For example, here. So these genes are on the same chromosome, then we can say they are linked. They're linked. But this is not enough, right? Because we know these 5 genes are on the same chromosome, but we also want to know how these 5 genes are arranged on this chromosome, right? And in other words, so we want to find out the positions of these 2 genes, and also we want to figure out the distance between these genes. So this practice is called the genetic mapping. So the mapping means like we will find the genetic map for those genes on the same chromosome. Excuse me. All right. So for mapping, for the gene map, actually, we have 2 versions. We have 2 versions of the map. So one version is called the physical map. So physical map, and the other version is called the linkage map or genetic map. So they're equivalent. When I say linkage map, so I can also say this is a genetic map. All right. So we have 2 versions of the map. The physical map is easy to understand because we use sequencing. We can so we can get the base pair information, right? So the physical map is a map that can measure the distance between 2 genes based on the base pair numbers. So for example, so if you look at this gene here and this gene here, so roughly the distance, the physical distance between these 2 genes are about maybe 500 base pair or something like that. So if we measure the distance between two genes using the physical base pairs. That's the physical map. If we, so for example, on this genetic map or linkage map, if we measure the distance between these two genes using another unit, which called the recombination frequency. So if we use the recombination, pay attention. So if we use the recombination frequency to measure the distance between any two genes, then that's the genetic map or the linkage map. So we have two versions. These two versions are actually related to each other, but they're not identical. So they're not exactly identical. They are related to each other. So the relationship between these two versions, the physical map or the genetic map, is very similar to the relationship between these two maps. So for example, when we talk about the city of London, so if we visit London, so probably you can get two free maps from the city. One is the physical map. Like this is very complicated. Sometimes like this map contains too much information for us to digest, right? So however, you can get another map for the same city but different format. So this map is for the subway map. Very clear. Just use the straight lines to link different stations, right? All right. So if you visit London, if you just want to go to one station from, you just want to go from one station to another station, I think the subway map will be more convenient for us to use, right? So, all right. So this is the same. So for example, when we talk about the genetic mapping, so maybe the physical map is too much information for us, but the genetic map is more convenient and useful for us to use to digest, to map the genes on the chromosome. So this is the relationship between two versions of the map. Okay, so all right. So when we talk, so genetic map. So today's topic is how we can derive the genetic map for genes on the same chromosome using the recombinations. See, so we're gonna try to associate these two things, right? So we want to map genes on the same chromosome. So how can we do that? So we can leverage the crossing over, the recombination between the two homologous chromosomes to help us derive the genetic map for the genes. So this is the topic for today. All right, so when we look at the chromosome, so for example here, if there's a crossing over occur, so there must be some DNA break, right? Breakage. So the DNA need to break first and then rejoin. All right. So let's understand how the DNA, the chromosome is gonna break. All right. So we can treat the chromosome like a chopstick. So this is a easy way to think about this. So if you have a chopstick, so if you order some Chinese food and you're done with the food and you're gonna throw them away, so you're gonna break the chopstick. So there are two possible ways for you to break the chopstick, use the chopstick. So first one, you can hold the two ends to break it, or the other way is you can hold in the middle of the chopstick to break it. So which one is better? Which one is better? Of course. So based on our experience, I think if you break the chopstick by holding on the two ends, that would be easier because the distance between this two holding position is far. So that means like you don't need to use a lot of like force to break it, right? This is physics, but we can use this principle to our crossing over the recombination between the two homologous chromosomes. So let's look at this example. So we have three genes here. So we have AB, we have D here. So the distance between B and D are very large, but the A and B are very closely related. So in this case, we say A and B are tightly linked. they are all linked. A, B, and D are linked genes because they are on the same chromosome. However, A and B are tightly linked because they are very close to each other. And the B and D and A and D are linked, weakly linked, right? Okay, so if there's a crossing over occurring between these two chromosomes, so it is more likely that the recombination or the crossing over will occur between B and D. Do you agree? Then in between A and B. So this is the principle we want to understand. So that's why I used the chopstick example to show you this principle. Okay. Now, so when we do a test cross, so because in midterm one, so we had a question that is related to the test cross, right? So yeah, that's the very important thing. In the next midterm two, we are also gonna we're gonna also have some questions that are related to test cross. So pay attention to here. So for example, if we do the mapping, so we also need to use test cross. So that means like everyone need to clearly understand what the test cross is, right? So here, if we, so for example, this is a hybrid, right? The tri-hybrid. So we're gonna test cross means like we're gonna cross this one with A, B, and D, right? Another A, B, and D. So the recessive, the 3 genes recessive. Okay, so we do this test cross, and okay, so that's the. Let me erase this kind of messy here. Okay. Okay, so when we do the test cross, we could get 2 groups of progeny, right? Two groups of the progeny. So one group is called the parental type. So now everybody knows what this means, right? Because this is important. The parental type means like the progeny carry exactly the genotypes like the parents. So the parents has a genotype like this, either the capital A, B, and D, or the lowercase a, B, and D. So in this group, if the progeny belongs to the parental type, you're gonna see they have exactly the identical genotypes like their parents, either the big ABD or the small ABD, right? And the majority of the progeny will be in this category. So because crossing over is actually it's relatively a rare situation. So that's why majority of the progeny will be the parental type. So this is very important. All right, now let's look at another category. So this category, we use R to represent. So this is the recombinant. Recombinant means like the progeny carrying different genotypes than their parents. Because of what? Because of the crossing over, right? So you're gonna see crossing over here, and here to result in this gamete and the progeny. All right, so now if you look into all the recombinant, you're gonna see a lot of crossing over occurring in between B and D. So those are all the progeny carrying the different genotype due to the crossing over between B and D, right? You're gonna see many of them are like this because B and D are the distance between B and D are very large. So the chance to have crossing over between B and D is very high. Okay, so of course you also can see something like this. So some progeny will have different genotypes, the non-parental genotype, due to the crossing over between A and B, right? But the number of this kind of like progeny recombinant will be very few, so fewer than this type. So I hope this is clear. So this is very important. All right. Okay, so now we're gonna use So in the future, we're going to use this test cross to help us to map genes. So this is a 2-gene model. So we start from the simplest model to make sure everybody understands this. And then we can easily extend this model to more complicated situation like the 3 genes mapping. Okay, so now when we talk about the 2 genes model, 2-gene model, so we try to map 2 genes. So we can use either the haploid model or the diploid model. So previously I asked you to read the textbook about the life cycle for the neurospora, right? So that's the very useful model organism, haploid model organism that we're going to use for genetic dissection, especially for mapping a gene. So we have one parent here, they're haploid, and when they come together to form this meiotic diploid cell, and after the meiosis, so you're going to see the progeny, right? So you're going to see the pool of progeny from this cross. All right, so when you compare the genotypes of this progeny with their parents, you're going to see, yeah, some of them are exactly identical to their parents. We say this parental type. And we see some other progeny are different from their parents. So here, we say the genotype, the parental genotype as the input, and those are the output. So we always compare the output genotype with the input genotype to figure out how many are the parental type, how many progeny are the recombinant. Recombinant means different from their parents, right? All right. So this is the haploid model. And in the diploid model, for example, the classical Mendelian pea plants experiment. So we start from 2 parents here. So the pure line, so those are inbred lines. And we so both the parents only have 2 genotypes, input genotypes, right? That's input genotype. And when we cross these 2 parents, we get the F1, which is the hybrid. So we are very familiar with this. And then we do the test cross. So this is test cross. We're going to cross this. This is the tester. And then we generate progeny like this. All right, the same. So this is the output from this test cross. We're going to compare the genotype of the output, the progeny genotypes with their parents. So as you can see, some of them are identical to their parents, but some of them are not. So the progeny carrying different genotypes than their parents are called the recombinants, right? Okay, so, all right, so I hope to remind you, so before today, so we talk about the independent genes, independently assorted genes. They are on different chromosomes. So in that case, you're gonna see the beautiful 1:1:1:1 ratio, right? Because of the independence between these 2 genes. And in that case, only 50%, for example, like this, 50% of the progeny will be recombinant, different from their parents. However, if the 2 genes are linked, like today, so the proportion of the recombinants will be less than 50%, right? So this is the criteria we can use to tell from a test cross. We can tell. So from a test cross, this is the criteria we can use to tell whether these 2 genes are linked or not. So the key point is this. We're going to use the 50% as a criteria, a cutoff, to test the linkage between 2 genes. Okay, so this is example here. So suppose this is the reality. Okay, so we have 2 genes located on the same chromosome. So we cross these 2 parents to generate the F1 hybrid, right? All right. Now, if we do the test cross, so if we do the test cross, So if these two genes, so here we can use this chi-square. So we can still use the chi-square test to test the linkage between two genes. We use the same. So before the midterm 1, we use chi-square to test the independence of the two genes, right? Actually, we can use the same test to test the linkage as well. Because here, if we want to test if these two genes are linked or not, we can test the non-hypothesis, which is, okay, these two genes are independent, okay? So this is the same, right? The same, the non-hypothesis. And the alternative hypothesis is, okay, these two genes are not independent, which means they are linked. I hope this is clear to you. Okay. Now, okay, when we test, when we put the non-hypothesis test on the trial, so if these two genes are linked, are independent, are unlinked, so this is the ratio we expect to see, right? This is exactly the same like before, like the midterm. All right. However, if you got the data like this, if this is your data, observed data. So what do you think? So in this case, if in next midterm, in the midterm 2, if I give you data like this, immediately you're gonna say, no, I don't believe these two genes are independent. They are linked. They must be linked because of the data like this, right? Okay, so all right, so let's look at the details about this. If we test across this one, this F1 individual with the double homozygous recessive individual, so we're gonna see if we see this data. So start this. Okay, so if you see data like this, the majority of the progeny will represent their parental type. So if you compare this and this, those two are parental types, right? All right, but if you look at here, those are different because they are different from their parental type. In the input genotype, there's no such genotype here. So that's the new combination of the alleles. All right, and you're gonna see much smaller numbers here. All right, that makes sense because crossing over is a rare situation. That's why. So for majority of the progeny, you do not see the crossing over. Then that's the parental type. But for a small portion of the progeny, you do see the recombinants. So that represent the recombinant. All right. So in the next midterm, so I'm gonna put something like this on the exam. So hopefully everyone can make it. All right. Okay, we use the same chi-square test to do that. Okay, so again, so this is a summary. So this is the linked gene. So if we do the test across for the linked gene, so we're gonna see the non-parental type, the recombinants will be significantly less than 50%. All right. So now I just put two mechanisms side by side. So this is the independent gene, the unlinked gene. This is the linked gene. So this is what we have learned before midterm 1. So if two genes are unlinked, if they are on different chromosomes, so you're gonna see this 50% of the progeny will be recombinants because these two genes are independent, right? However, if these two genes are not independent, if they're linked, you're gonna see less than 50% of the progeny will be the recombinants. So this is today's topic. This is the difference between today and what we have learned before. I hope this is clear to everyone. So we start from a very simple model case, and now we little by little we just get a little bit complicated situation, but we use the same principles, rules to help us to do the genetic analysis. Okay, so this slide just show you the evidence of the crossing over. All right, so this is a picture taken, I believe, from the microscope. So as you can see during the meiosis, there's like crossing over occurring here and here. So yeah, and this is another mechanism that can produce a recombination. So in the midterm, I asked the question, so what is recombination? So and what mechanism can cause the recombination? So there are two possible ways to answer this. One is the independent assortment, so that can produce new genotypes when compared to the parents, right? And also the crossing over between two homologous chromosomes can also produce recombinants. Right. So of course, there are more mechanism can produce recombinants, but those two are the most commonly seen mechanism for producing recombinants. Okay, now, so as I mentioned, today we're going to learn how we can use the crossing over or use the recombination frequency to derive the genetic map for genes, right? So which means like how we can use the crossing over information to measure the distance between two genes on the same chromosome. So for example, so we have two genes on the same chromosome. So how can we use the recombination between these two genes, the information of the recombination between these two genes to help us to measure the distance, genetic distance between these two genes? So this is the model we're going to use. So we're going to calculate the genetic distance between two genes using the recombination frequency, or non-parental. NP means non-parental progeny frequency. All right. This the unit for the genetic map distance, or the linkage distance is the centimorgan, or the map unit. All right, they're equivalent. So when I say 1 centimorgan, so it is equivalent to 1 map unit. And 1 map unit or 1 centimorgan is equivalent to the 1% observed recombination. Or in another way is, okay, so if you put the total number of the progeny, this is the progeny, okay, progeny here. So in a test cross, you know how many, you know the sample size, right? So the sample size, the total sample of the progeny, total number of the progeny will be the sample size. That total number of the progeny, you're going to put this number in the denominator, and in the numerator, you're going to see out of out of all the progeny, how many represent the recombinants, the number of recombinants. Okay, sorry. Okay, all right. And this ratio actually can help you, can be translated into the map distance between these two genes, right? And the larger number here refers to the further, so the further away they are on the chromosome, the larger this number will be, the map distance. That makes sense, right? And of course, the maximum genetic distance between any two genes will be 50 centimorgan. So why is that? Why is that? Because think about, like, if these two genes are unlinked, if these two genes are independently assorted, so the percentage of the recombinants from that cross will be 50%, right? So we just learned this before here. So that's the maximum genetic distance between any two genes if they are linked. So for any genetic distance between two genes, this measure will be less than 50 centimorgan, right? Okay, so this slide actually just shows you the mechanism, biochemical, the genetic mechanism behind the crossing over. So this is not required for the class, but if you have time, so please read the textbook to make sure you understand what is going on behind the crossing over between two homologous chromosomes during the meiosis. All right. Okay. All right. Now, this is very, very important. All right. Because as I mentioned, we need to calculate the genetic distance, right? The 2G model. So this is a 2G model, simplest. So if we want to calculate the genetic distance between these two genes, we need to do the test cross first. We need to do the test cross. We need to generate a lot of progeny from the test cross. And then we have the total number of the progeny here. So this is your sample size. For example, if you generate like 1,000 progeny, and from this 1,000, you're gonna see how many of them are recombinant, non-parental type. How many of them carry the genotype that are different from their parents, right? All right, this ratio will give you the genetic distance between these two genes. So let me give you an example. So in the test, yeah, I could give you something like this, right? We have a this parent is the dihybrid parent. So we just cross this parent with the double homozygous, the homozygous recessive tester, and we generate this, right? So if these two genes are independent, so we're gonna see 1 to 1 to 1 to 1, right? If they are not, so we're gonna see a different ratios. So this is different ratios here. Okay, so from this output, you're gonna see some categories are very, so a lot of like progeny. So you're gonna see majority of the progeny carry the parental type here, and only a small proportion of the progeny carried non-parental type. Because this is a 2G model, simple model, so we immediately know this non-parental type is due to the crossing over, right? So because these two genes cannot be unlinked, they are linked. That's why you're going to see a ratio that is not 1 to 1 to 1 to 1. However, so when you check into these 4 categories, you're going to see the small number of the progeny will represent the non-parental type. So this is the number you need to pay attention, right? So so the total number is 100. So we generated this many progeny. Total number is 100, and out of this 100, we have this many non-parental type recombinant. And we simply calculate the ratio of this, and this gives you 10%. So that means like the recombination frequency between these two genes, B and L, will be 10%. This will translate to a 10 map unit or 10 centimorgan between B and L, right? So if you draw a diagram here, so this is one chromosome, and this is B, and this is L gene, and the distance between them should be 10 map units. So this is how we do the calculation. Very important. Yeah. Again, very important. So I hope everyone knows how to do the calculation, because this will be something on your quiz and also on the second midterm. Right. Okay, so map distance, actually, there's some features for the map distance. Usually they are additive. So what does that mean, additive, the map distance? So for example, so if we do the test cross to figure out the map distance between A and B, there's 5 map unit. And we do the same thing to measure the distance between A and C, that gives you 3 map unit. And if these three genes are arranged in this way, so A is in the middle, and this is C and this is B, then the distance between the genetic distance between C and B will be the sum of these two numbers, which is the 8 map unit. So this feature means the additive feature of the map distance. All right. So if the three genes are arranged in this way, if gene C is in the middle, so of course, so you're gonna have this relationship between the three genes. Right. Okay, so now, so as I mentioned, so this is 2-gene model. This is 2-gene model, the simple one. So so if we fully understand this, so we're gonna challenge ourselves. So we're gonna deal with some more difficult situations. So here we try to expand this model to the 3-point test cross, now 3-gene model. So here we just test cross this tri-hybrid. Okay, so we have a tri-hybrid parent, and this is the tester. So homozygous recessive for 3 genes, right? All right. So there's one possible way. So this is the majority progeny from this cross. So you're going to see the parental type. So there's no crossing over between at these 2 chromosomes. You're going to see you're going to see the parental type here, right? All right. This is the parental type. This is also the parental type. Okay. Oops. This is the parental type. Okay. All right. Now, if there's a single crossing over occurring between A and B, so for example, there's a crossing over occurring here. So you're going to generate this recombinant and this recombinant. Makes sense, right? There's another way to produce recombinant. So if the crossing over occurring between B and C rather than A and B, so you're going to generate a little bit different recombinant progeny, non-parental progeny, right? And let's look at the last example. So this is a little bit tricky here, because in this case, if we have double crossing over, so one occurring between A and B, and the other one is occurring between B and C. So you're going to generate something like this. So those are, of course, they are recombinant, but the interesting thing is, like, if you compare this progeny with their parents, so with the parental, so if you compare this genotype, the non-parental genotype, with this parental genotype, so only the genes in the middle has been swapped, switched, right? So this is, yeah, this is the thing. Yeah, I hope everyone can pay attention. So if you compare the progeny of double crossing over, you're gonna, so if you compare the double crossing over progeny versus the parental type, you're gonna see only the gene in the middle gene will be swapped because of the double crossover, right? Okay. All right, let's look at the example. Okay, so here, so because we want to use this to do what? So I hope, yeah, everybody still a bit clear. So we try to use this crossing overs, the test crosses, and we're gonna generate the crossing over frequency to help us map the 3 genes, right? We want to find out the arrangement of the 3 genes on the same chromosome, and then we want to find the distance, genetic distance between any 2 of them, right? Because right now we do not know this information, so we just put dot here. So we know these 3 genes are linked, but we don't know how they are linked and what's the distance between these 3 genes. So we start from 2 inbred lines here, 2 parental type lines to generate the tri-hybrid here. And we're gonna test cross this one with the homozygous recessive to generate a F2 population. So this is the F2 progeny. So we're gonna analyze this F2 progeny to help us figure out the genetic map for these 3 genes, right? Okay, so in the exam, I could give you a question like this. So, all right, so we start from the two parental lines, and we cross these two parental lines to create the F1 tri-hybrid here, and then we do the test cross, right? Test cross, and then we produce, this is the progeny, F2 progeny, right? All right, so wow, what a messy table. So how can we deal with this table? So let me show you how we can analyze this table. And once you got this, so there will be no problem for the midterm and quiz. Let me clear the slides first. Okay. So yes, this is a 3-gene model. So it's a little bit complicated than the 2-gene model, but we still use some similar principles to help us to do the genetic dissection. Look at here. So those are the numbers in each different category, right? So if we look at these numbers, these two numbers are way more larger than the other categories. So what do you think? Based on the information we just talked about. Because the crossing over will cost energy. So that's a relatively rare situation. So if you see the big numbers in this table, this immediately tell you, okay, they must be the parental type. So the parental type must be like this, right? And okay, so out of this column, so you're gonna see two numbers are very small, extremely small compared to other numbers. So what does that mean? So because the one crossing over is rare, how about two crossing over, the double crossover? So that's even rarer, right? Much rarer. So that category will represent the double crossing over, the recombinant progeny due to the double crossover. All right, so now we have the parental type. We have the double crossing over. So as I mentioned, if you compare the double crossing over progeny versus the parental type, the only difference is the gene in the middle will be swapped, right? So this is what we can use, this information. Okay, so now so if you compare this parental type with this double cross over progeny, you're gonna see this one is the same. This one is the same. And only this one is different. So what information can you get from this comparison? This means, okay, these three genes must be arranged in a way that the CT will sit in the middle, right? Immediately you can get this information. I hope this is clear, right? And okay, now, so because this is a 3-gene model, so it's difficult because we only have the formula to calculate the distance between two genes, 2-gene model, right? So how can we do that? It's not that hard. So we can treat this 3 genes. We can divide this 3-gene problem into 2-gene problem as well, right? So we can calculate the distance between any pair of these 3 genes. So for example, if we just look at the V and CV, so we can use the 2-gene model to help us to calculate the distance, genetic distance between V and CV, right? So out of these 8 categories, this represents the recombinant progeny for these 2 genes. So look at here, right? So the parental type is so if we look at the V and CV, so V and CV, the parental type is V CV plus or V plus CV. So if you look at any of these 4, so this will gives you the recombinants for this 2 genes, right? Those are the recombinants, and those are the recombinants. So first, when we look at these 2 genes, if we want to use 2-gene model to calculate the distance between these 2 genes, we need to figure out which category represent the recombinants in terms of these 2 genes. The same thing, so when we look at this gene pair, so we're going to figure out the recombinant progeny for these 2 genes, and the same for this, right? Okay, now when we have this information, we can easily calculate the genetic distance. So we have learned the 2-gene model. So what is the genetic distance between this gene and this gene? Because those 4 represent the recombinants for this 2 genes, and so we only use these 4 numbers here. So you add up these 4 numbers will become this number, and the total number is this. This will be the denominator, and this 4 number will add up to this number in the numerator. So the genetic distance between V and CV should be this. This is very simple, right? It's not that hard. All right, so you can repeatedly do this for this 2 gene. Because when you look at these 2 genes, so this 4 will represent the recombinants. So you're gonna use these 4 numbers here, and you add these 4 numbers to get these numbers here, and you can get the genetic map distance between these 2 genes. And the same, so if you look at these 2 genes, these 4 categories and this 4 numbers will add up to this, and this will give you the genetic map between these 2 genes, right? Okay, now you can put them together. So it's very, very easy. So the V and the CT is equal to this 13.2 map unit, and the distance between CT and CV is 6.4. But the problem is like, okay, so professor, you just said, okay, so the map distance will be additive. So why these 2 numbers do not add up to these numbers, right? So why they do not add up to these numbers? So let me tell you why. All right. So if you use this table, if you use this table to summarize the results, the progeny produced from this test cross. All right, so the first 2 numbers represent the parental type, right? The non-crossover. So we can say non-crossover here. All right, so if we knew the city is in the middle. So we define this region as 1, this region as 1, and this region as 2. All right, so because for the parental type, there's no crossing over at all. So we put none here. All right, when we look at these 2 categories, this represented a single crossover, but in region 2. These 2 categories. All right, now if you look at these 2 categories here, this represent single crossover as well, but in different region 1. So there's a single crossover in region 1 will produce this kind of progeny. However, when you look at the last category, so if we look at these 2 categories with the smallest numbers, this represents a double crossing over, which means each one will have 2 crossover. One is in the region 1, and the other one is in the region 2. So when we do the 2-gene model genetic mapping calculation, we did not consider the double crossing over. We just count these individuals, these numbers only 1 time, right? Actually, we need to double their size. We need to count them twice because they have 2 crossing overs occurring in the meiosis to produce the progeny like this. Because remember, the map distance in 2-gene model, the map distance is defined as the recombination frequency. So those are the double crossing over. Their numbers should be multiplied by 2 to represent the real crossing over numbers for this progeny. Okay, let's do this correction. See what happens here. All right, for the first one, it's easy. If we want to calculate the V, let's say V and CT, so that's easy. That's the same calculated number like we did before. So if we calculate the distance between CT and CV, the same here. However, when we calculate the distance between V and CV, so we need to multiply the number of double crossing over progeny by 2. So if we do that, then what's the result? It will end up with the right genetic distance between these two genes, CV. Because yeah, we need to do this correction, but in reality, so this slide just show you how we do the problem. So in reality, so in the exam or in the quiz, you do not have to calculate the largest number, right? You do not need to do this correction because that can take extra time. But because we knew genetic maps are additive, so once you get the three numbers, so you just the two small numbers will be correct. So you will accurately calculate the two small numbers, and then you just simply add up these two small numbers to result in the largest genetic map between the two genes that are far removed from each other. So you do not have to do the correction like I did here, because that wasted of time, because this will give you the same answer. So the easiest way is like here. Erase this. So in the test, the easiest way for you to calculate the genetic map distance is, okay, so you follow this 2-gene model to calculate 3 genetic maps distance, right? And you put them in the right order, like this. And you put the small numbers here, and you do not have to do the correction. You just add up these two numbers to give me the largest map distance, which is the 19.6. I hope this is clear. Okay. All right. So during the test, during the exam, so I may give you a question like this. So it will be helpful for you to use this table to summarize this cross. All right, because here this is so the first step is like, so the first step is always like this. This will be the easiest way. So from this output, you can immediately find out the parental type, right? The parental type represents the category with the largest numbers, no doubt, right? Okay, and also you can immediately identify the double crossing over progeny because they represent the smallest group, right? So once you got this two, so you compare the genotype of the double crossing over with the parental type, you can immediately see which one is which one is changed, and that one must be standing in the middle. Okay. All right, because without this information, you do not know which one is standing in the middle, right? There are 3 possible ways to do this. So either the CT is in the middle, or the V is in the middle, or the CV is in the middle. All of them are equally likely. However, if we got the data like this information will immediately rule out this possibility, and it gives you the right answer here. So this is the first step. So this, and then you just follow the, let me see. Oops. What is this? Okay, so so in the test, so once you got this results, then you can get the full credit for this problem, because here we do not, for this mapping, so we just try to map the 3 genes on the same chromosome, right? This mapping does not involve the centromere. So in this case, you just put the 3 genes in this way or in another way. As long as you put the CT in the middle, yeah, that's the correct answer. You can, so if you give me this results or another results, with the CT in the middle, but CV on the left and V on the right, that's okay. So they are equally good. So you can get the credit. So because in this case, we do not involve the central mirror in the mapping. But in the future, when we use the neurospora as a model organism, so in that mapping strategy, if we involve the central mirror in the mapping, so that's a little bit different. That's more complicated, but yeah, we're gonna learn that in the future, but not today. And today, this is what we need to learn. So let me give you a summary. So today we talk about a quite different topic, right? So before the midterm one, we just focus on two independent genes. So why do we say they are independent? Because those two genes in the past, those two genes are located on two different chromosomes. All right, if we do the test cross, we always produce 50% of the progeny representing recombinants, right? Okay. Now today we focus on different situations. So we're talking about genes are linked. They are located on the same chromosome. So if that is true, all right, then we do the test cross. We're gonna see the recombinant progeny, but the proportion of the recombinant progeny from the test cross will be significantly less than 50%. Because of what? Because the crossover is not a free lunch. It costs energy. So that's why it's a rare situation. That's why in the majority of the progeny, you're going to see the parental type. Only in a small portion of the progeny will show the non-parental type. So this is the major difference between today and the previous. And we also use the basic two-gene model to show how we can leverage the test cross to help us to measure the genetic distance between these two genes using the recombination frequency, right? So we from the test cross, we can produce a lot of progeny. And we just check what's the proportion of the progeny, the total progeny will have the non-parental type. So that ratio gives us the calculation of the genetic map distance between the two genes. All right, that's the easiest model. We can expand this model to 3 genes. So then we can easily map 3 genes to see how these 3 genes are arranged on the chromosome. This is the table I just showed you, right? So we can use this table to map the 3 genes. And there's a strategy for doing this. So I hope, I make it clear. So, and all the strategies are make sense to you. So make sure you understand this, because this is very important. And I'm gonna give you some questions in the next midterm to let you to calculate genetic distance between 3 genes using data like this, right? Okay, so I think we're doing good today. So I think I'm gonna stop here. So if you have any questions, I can help answer for today's. Any questions? Good. You don't. Yeah, you do not have any questions. That means like everybody doing good. Yeah. So actually today's content is very important. It is very basic, but it's a very important principle. We're gonna repeatedly use this in the future. Oops, I saw some like questions. Slides 20. How to get, okay, 20, slides 20. Sure, let me see. Slides, so sorry, sometimes I did not pay too much attention. on the chat box. So, all right, sorry about that. Let me see. 20 here. Okay, sure. So let me see. So how do we specify R for each gene pair, right? Okay, so of course, so first we know this is the parental type, right? The parental type means like if we look at these two genes, so the parental type will be V, either V CV plus or V plus CV, right? So I hope you agree with this. So either this or this. Okay, now out of this eight categories, so we want to see which one represents the recombinants for these two genes. So let's look at here. So in this one, it's V and CV. So that's different from parental type, right? So of course we put a R here. So that's the recombinant. If you look at here, so this is V plus and CV plus. That's different from the parental type. Also R. Okay, the same for this one and this one. That's why we put 4 R's here. Okay, but if you look at here, they are identical to the parental type. So that's why they are not. So for this two genes, we specify 4 recombinant categories. So doing the same thing, we can identify the recombinants for these two genes as well. Is that clear? Okay, any other questions? Let me see the chat box. Oops. Okay. All right. Okay, sure, thank you. Any other questions? Let's see. Good. Yeah, it looks like, yeah, today's lecture is so it's good. So I hope like everything is clear. So if anything is not clear, just send me an email. So I will be very happy to explain anything by email to you. All right. So let me stop sharing here. All right. Any other questions I can help for today? Yeah, if not, I think we can dismiss the class a little bit earlier today. All right. Thank you, everyone. Have a good one. Bye-bye.